Welcome to Simplified UPSC by PPP, where we listen, learn, and grow together. Detailed lecture notes in English and Hindi, plus practice questions for prelims and mains, only on our Telegram channel. Link in description. Like and subscribe for more.
Welcome back to the deep dive. Our mission here is, well, it's pretty simple. We take complex foundational material and we distill it into the high-impact knowledge you need.
Especially if you're navigating those high-stakes competitive exams.
Exactly. And today's deep dive is a foundational master summary. It's all drawn directly from core Class 12 biology concepts.
And this is critical stuff. This is the groundwork for understanding, everything from modern medicine to global environmental policy.
Right. Our objective is to pull out the essential facts. But more than that, to find the connective tissue, the links between genetics, biotech, human welfare, and ecology.
What's so fascinating here is that we aren't just, reading chapters from a textbook.
Yeah.
We're showing how the laws that govern a simple pea plant's inheritance directly inform crop breeding strategies, how understanding DNA dictates pollution policy. It's all about the so what.
And before we begin this pretty dense journey, I just want to say a heartfelt thank you to every single one of our existing members and subscribers.
You really mean that?
We do. Your support is what powers us to create this detailed content. So if you're finding this useful, please consider hitting that like button, subscribe, share your comments. And if you're able, we humbly ask you to consider joining our paid membership. It helps ensure we can keep delivering these deep dives.
Okay, let's unpack this.
So we have to start at the beginning. The foundational rules of inheritance from Gregor Mendel.
Of course.
But instead of just listing the laws, what do they really let us predict about how traits are passed down?
They gave us a blueprint, a predictable one. The law of dominance, for instance, explains why if you cross two different true breeding parents, only one trait, the dominant one, shows up in that first generation.
But the really revolutionary ideas were segregation and independent assortment.
Absolutely. The law of segregation. That's the one where traits don't blend, right?
Precisely. It tells you that the characters, the alleles, they separate cleanly during gamete formation. They aren't diluted or mixed.
That separation is key.
It is. And independent assortment takes it a step further. It says when you're looking at two different characters, say, seed color and seed shape,
the factors for each trait mix and match completely freely.
they combine in every possible way. That's what makes complex breeding possible.
And it's so important to remember the difference between the unseen, the genotype,
The actual genetic code.
and the visible result, the phenotype. But to really understand how those factors work, we have to look inside the cell, at DNA.
For decades, scientists just weren't sure. Was it DNA or was it protein that held the code?
That started to change with Griffith's experiment, a famous one. He showed that something, he called it the transforming principle, could jump from dead, virulent bacteria,
The S strain.
to live, harmless bacteria, the R strain, and turn that R strain virulent. It meant genetic information was physically being transferred.
But the big question was still there. What was that substance?
And that was answered decisively by Avery, McLeod, and McCarty. Their brilliant step was purifying the components, DNA, RNA, and protein, from those heat-killed S cells.
So they isolated each one.
They did, and they found that only the presence of DNA caused the transformation. And here's the crucial part. When they added an enzyme called DNase, which chews up DNA,
The transformation just stopped.
it stopped completely. That single step proved, really without a doubt, that DNA was the transforming substance.
Now let's jump to the mega scale. The Human Genome Project launched in 1990.
An incredible project, and the numbers are just staggering. 3,164.7 million base pairs sequenced.
And what did those numbers tell us?
Well, beyond just the length, we learned the average gene is about 3,000 bases. But you see huge variations. The largest known gene, dystrophin, is 2.4 million bases long.
Wow.
It shows the complexity. But maybe the most profound takeaway is that 99.9% of our DNA sequences are identical across all humans.
So all our incredible diversity comes from that tiny 0.1%.
Exactly. It speaks volumes about our shared genetic heritage.
And that understanding of the blueprint, it translates directly into the real world. This is where it gets really interesting. Let's connect that knowledge of genetics to the Green Revolution in India. We learned how to manipulate and breed crops for higher yields.
The Green Revolution was a global triumph of plant breeding. In India, it was spearheaded by M.S. Swaminathan, building on Norman Borlaug's work.
And the goal wasn't just higher yield, was it?
No, it was also stability, creating varieties that were high-yielding and disease-resistant.
The key idea was semi-dwarfism. By introducing genes for a shorter stature, the plants didn't waste energy growing tall,
Right. They funneled that energy directly into the grain.
increasing the harvest. You just have to think of the impact of those semi-dwarf wheat varieties, Sonolica and Calliansona.
And later, the rice derivatives like Jaya and Ratna, they changed everything.
And while we're on food, we should mention the Blue Revolution.
Yes, the massive increase in production from aquatic sources. Fish, plants. It's vital for coastal economies and global protein supply.
Okay, let's shift from food to health. Recombinant DNA technology has been revolutionary.
It really has. We're talking about genetically modified organisms, GMOs, where we've altered the genes.
And a prime example is genetically engineered insulin. Before this, insulin came from animals, from pigs and cattle.
And it often caused severe allergic reactions. The human body saw that animal protein as foreign.
So what did genetic engineering change?
Well, because we now understand the human gene sequence, we can use biotechnology to create human insulin that is a perfect match to our natural molecule.
So this recombinant insulin.
It avoids those immunological conflicts entirely. It's made treatment safe and effective for millions.
Speaking of safety, let's touch on immunity. Vaccination works entirely on the basis of immunological memory, right?
Exactly. When you introduce a harmless version of a pathogen, the immune system doesn't just fight it off, it creates memory B and T cells.
They're like a highly trained unit waiting in reserve.
That's a great way to put it. If the real pathogen ever shows up, those memory cells mobilize so quickly they overwhelm it, often before you even feel sick.
But biology is also transforming basic farming away from high-tech labs through things like biocontrol.
Yes, using one biological agent to control a pest, instead of just spraying toxic chemicals.
It's a move toward integrated organic farming.
A closed-loop system. For example, using the fungus trichoderma to fight plant pathogens in the soil, or using baculoviruses to specifically target insects without harming other beneficial species.
Like that model from Ramesh Chandra Duggar.
Exactly. He even uses cattle waste to create natural gas. It reduces the reliance on expensive and harmful agrochemicals. It's a whole system.
All right, let's shift our focus to ecology, how organisms interact with their environment.
How they cope with change.
We tend to group them into two types, regulators and conformers.
Right. Regulators, like us mammals, maintain a constant internal environment. We keep our bodies at a steady 37 degrees Celsius.
Which takes a lot of energy for sweating, for shivering.
It does. Conformers, on the other hand, just let their internal state match the outside temperature.
When things get really harsh.
They use behavioral strategies. Some snails enter estivation to survive a hot summer, while some zooplankton enter diapause, a state of suspended development when conditions are bad.
We see these interactions play out in populations too. Predation, for example. It often gets a bad rap.
It does.
But it's actually critical. It's nature's way of transferring energy and, more importantly, maintaining species diversity. It stops one or two prey species from just taking over everything.
The classic example is the pisaster starfish.
Tell us about that.
When this top predator was removed from its intertidal habitat, interspecies competition just exploded.
And the result?
Over 10 other invertebrate species vanished from that area. It proves that predation is essential for ecological stability.
And you have these other specialized relationships, like parasitism.
Where one benefits at the other's expense. It can be ectoparasites on the surface, like lice, or endoparasites living deep inside organs.
And on the other end of the spectrum, you have mutualism.
The perfect two-way street where both partners benefit. The relationship between a specific fig species and its partner wasp is just a stunning example of coevolution.
The plant needs the wasp for pollination.
And the wasp relies entirely on that plant for its larval development. They can't live without each other.
Connecting this to the bigger picture, the need for biodiversity conservation is, well, it's urgent.
It is. We're facing extinction rates that are estimated to be 100 to 1,000 times higher than in past mass extinctions.
So how do we combat that loss? We focus on biodiversity hotspots.
These are regions with incredibly high species richness and a large degree of endemism.
Meaning species found nowhere else on Earth.
Exactly. The global map has 34 of these hotspots, and 3 of them cover parts of India. The Western Ghats and Sri Lanka, Indo-Burma, and the Himalaya.
And conservation strategies fall into 2 main baskets, right? In situ and ex situ?
Right. In situ is protecting the ecosystem in its natural habitat. Think of India's 14 biosphere reserves and hundreds of national parks.
And ex situ.
That's protecting species off-site. in zoological parks or botanical gardens.
What's often forgotten, though, are the powerful community actions, not just government policy.
Absolutely. Policy can be huge, like the Supreme Court mandating Delhi's public transport switch to compressed natural gas or CNG by 2002.
Why was CNG so effective?
It's more efficient, it's cheaper, and crucially, it's much harder for vendors to adulterate than petrol or diesel.
But local innovation can be just as impactful. Look at Ahmed Khan in Bangalore.
A fantastic story. He solved a huge plastic waste problem by developing Polyblend.
It's a recycled plastic powder.
Yes, and when you mix it with bitumen for road construction, it actually enhances the road's water-repellent properties. It provides a valuable, profitable use for plastic waste and makes the roads last longer.
Now, what about the ethical dimension of all this, especially around biological resources and traditional knowledge?
You're talking about biopiracy?
Exactly. Let's define it. It's the unauthorized use of bioresources by, say, multinational companies without proper authorization or payment to the community that holds that traditional knowledge.
And the most cited example involving India is the basmati rice case.
What happened there?
An American company received a patent in 1997 on a new variety that was derived directly from traditional Indian farmer varieties. It was essentially trying to patent centuries of knowledge.
So to manage these new challenges, the Indian government established GEAC.
The Genetic Engineering Approval Committee, its job is to evaluate the validity and the safety of all research involving GMOs.
Shifting to a global scale, we face the threat of climate change, driven by greenhouse gases like carbon dioxide and methane.
Yes, contributing to global warming. But we also have to deal with the degradation of the stratospheric good ozone layer.
The one that protects us from harmful UV radiation.
That's the one. It's depleted primarily by chlorofluorocarbons, or CFCs. When CFCs get up into the stratosphere, UV rays release chlorine atoms,
And those atoms act as catalysts.
relentlessly breaking down ozone molecules. The increased UV exposure leads to serious risks: skin cancer, snow blindness.
And the international community's response was the Montreal Protocol.
In 1987, an agreement to control and eventually phase out those ozone-depleting substances.
So we've completed a pretty wide-ranging deep dive here. We've linked the smallest components, the genetic laws, to the largest global challenges like conservation and climate policy.
This foundational, interconnected approach, it's really essential for true mastery of these topics.
It is.
And, we highlighted the incredible conservation effort by the Bishnoi community back in 1731, led by Amrita Devi. They sacrificed hundreds of lives to save trees.
An unbelievable story of commitment.
It is, and it raises an important question for you to reflect on. When environmental threats accelerate and biopiracy remains a constant risk, are our centralized regulatory frameworks like the Montreal Protocol or national bodies like GAC, are they truly sufficient? Or must ethical, community-based values and traditional knowledge start to play a much larger, legally binding role in how we use and conserve our resources moving forward?
That is a powerful thought to consider. Thank you again to all of our dedicated members for making this work possible. We really encourage you to keep digging deeper into these vital topics.
Please remember to subscribe, like, and share.
We'll be back soon with more high-yield knowledge.
Thanks for watching. Follow us on Telegram and Instagram. Don't forget to like, share, and subscribe.